This section is about mutations, the M word that I keep talking about. Mutations are changes in the DNA. Comes from a Latin word, like we do in science, in biology specifically. These are able to be passed down. They're inherited changes in genetic information. There are two main categories of mutations: single gene mutations and chromosomal mutations. We're going to go through both. Mutations that affect one or a few nucleotides are called point mutations because they occur at a single point in the DNA. This is a copying error during replication. If a gene in one cell is altered, that alteration can be passed on to every cell that develops from that original one. There are three types of point mutations: substitutions, insertions, and deletions. And lucky for us, they're exactly like they sound. In a substitution, one base is changed to a different base. Generally speaking, substitutions affect no more than a single amino acid. Sometimes they don't even have an effect. So what you see going on here is the normal sequence is here. What has happened is, instead of there being a GCA here, it's GTA. And then through the processes of transcription and translation, the amino acid has changed from arginine to histidine. Which is pictured here. A change in the last base of that codon, though, from CGU to CGA, would still specify that same amino acid, arginine. So sometimes substitutions don't have any effect. Insertions and deletions are more serious. This is where one base is either inserted or removed from the DNA sequence, just like they sound. If a nucleotide is added or deleted, the bases are still in codons, groups of 3, but now everything has shifted down after that insertion. In this case, this A has been inserted. So what has happened is all 3 of these amino acids, and then on down the road also, will all change. Here, this G has been deleted. So it shifts everything back. And again, all of these, as well as all the amino acids after them, will change. These types of insertions and deletions are called frame shift mutations because they shift what we call the reading frame. The codons are shifted over by one base. They can, like I showed, potentially change every amino acid that follows the point of the mutation and can alter a protein so that it's unrecognizable and the cell will just destroy it and say this is defective. Chromosomal mutations are just like they sound. They involve changes in number or structure of chromosomes. They can change the locations of genes, change the number of copies of some genes. There are 4 types of chromosomal mutations. And again, lucky for us, they sound exactly like what they are: deletion, duplication, inversion, and translocation. Deletion involves the loss of all or part of a chromosome. So in this case, section B has been eliminated from the arm of that chromosome. Duplication produces an extra copy. So for some reason, 2 sections B are involved in this chromosome. When you invert something, you turn it upside down. So with an inversion, what you see here is that sections B and C have been flipped around. And then translocation occurs when part of one chromosome breaks off and attaches to another. So GH is from another completely different chromosome. It inserts itself into the original chromosome. Alterations like this can happen naturally or artificially. We'll talk about artificial alterations next set of chapters, which is genetic engineering. These mutations can affect an organism or maybe not. Some mutations that affect individual organisms can also affect an entire species or even an entire ecosystem. Lots of mutations are produced by errors in genetic processes. Like I said before, point mutations are basically DNA copy errors, errors in DNA replication. The cellular machinery, however, is pretty accurate. It inserts an incorrect base roughly once in every 10 million bases. Small changes in genes can accumulate slowly over time. When we're talking about bacteria, they have a very short generation time. Environmental conditions can cause bacteria to increase their mutations. And that can actually be helpful. Mutations can give bacteria new traits, such as the ability to consume a new food source or resist a poison or an antibiotic. Some mutations arise from what we call mutagens. These are chemicals or physical agents in the environment. Some examples include pesticides, plant alkaloids, tobacco smoke, and environmental pollutants. These are chemical mutagens. Physical mutagens, electromagnetic radiation like X-rays and ultraviolet light. If these mutagens get into contact with DNA, they produce mutations at high rates. Some compounds interfere with base pairing, increasing the error rate of replication. Some weaken DNA, causing breaks and inversions that produce mutations. There's a repair mechanism in place. A lot of times DNA can be repaired when there are mutations, but when they cannot, there's a permanent change that results. You've asked me before, are there helpful mutations? Absolutely. We talk mostly about the harmful mutations, but the effects can vary widely. Some are good, some are no effect, and some are bad. To determine whether a mutation is negative or beneficial, you got to see how its DNA changes relative to the situation. Most of the time, like I said, we think of mutations as being negative because they disrupt the normal function of genes. But without mutations, there can't be change over time. Mutations cause genetic variation in a species, which is one of the awesome things about reproduction in sexually reproducing organisms. Some of the most harmful mutations can change protein structure or gene activity. The defective proteins can disrupt normal biological activities. Building things up, breaking things down aren't done, and the buildup of these products can be toxic. That results in genetic disorders. Some cancers are the product of mutations that cause uncontrolled cell growth. Sickle cell disease is a disorder associated with that moon-shaped cell. It's caused by a point mutation in one of the polypeptides, the proteins found in hemoglobin. Symptoms can include anemia, severe pain, frequent infections, and stunted growth. We also saw that having sickle cell trait can protect a person against malaria. So there are beneficial effects to some of these. Some mutations have helped insects resist chemical pesticides. Some mutations have enabled microorganisms, bacteria, to become antibiotic resistant. Neither of these is necessarily good for human society, but it's good for these species. Plant and animal breeders try to take advantage of good mutations. In plants, when a complete set of chromosomes fails to separate during meiosis, the gametes may produce triploid or tetraploid organisms. This is called polyploidy, and there's an extra set of chromosomes. Polyploid plants, say that 3 times fast, are often larger and stronger than diploid plants. Bananas and limes have been produced in this way. These are really important crop plants. Polyploidy also occurs naturally in citrus plants through spontaneous mutation.